Welcome back, everyone. Happy spring 2023 semester. We're very excited for the speakers we've got lined up for you. Before I begin this introduction, though, I wanted to just let you all know that next Monday on February 6th, our speaker will be in person in PH 2200. So if you are able to attend in person, please, by all means, do try and make it. There will also be the hybrid option as usual, but we just wanted to put that out there and let you all know. But back to today, we are very excited to welcome our first speaker for 2023. So Dr. Leonardo Tozzi, MD, PhD, graduated as a medical doctor from Pisa University and Sant'Anna School of Advanced Studies in 2013. In 2018, he was awarded his PhD from Trinity College Dublin for his research on the impact of genetics, epigenetics, and environmental stressors on structural and functional brain changes related to depression. Leonardo joined Stanford University in 2018 as the postdoctoral lead of the Human Connectome Project for disordered emotional states. Since 2022, he leads the Computational Neuroscience and Neuroimaging program at the Stanford Center for Precision Mental Health and Wellness. The goal of Leonardo's research is to develop quantitative biomarkers for mood disorders that are reliable, interpretable, and can be used to guide treatment selection and estimate treatment response. To this end, he integrates large-scale recordings of brain structure and function with behavioral measures and symptoms, as well as other biological markers. And with that, I will let Dr. Tozzi take it away. Thank you so much for being here today.
Thank you so much for inviting me. It's really a pleasure and an honor to be here. And thank you very much for the nice introduction. I am sharing my slides now. So, just like, Courtney said in the introduction, my work at the Stanford Center for Precision Mental Health and Wellness is really about finding a way to give psychiatry a quantitative edge, so to speak. As you know, in psychiatry, compared to other branches of medicine, it's rather uncommon to have biological markers or biomarkers to guide treatment decisions or prognostics. So what we're trying to do really is to use brain scans to inform, for example, what medication a patient should take rather than, what behavioral treatment a person should take, and so on and so forth. So because I think like most of you or many of you are not really into brain imaging in psychiatry, which is kind of a niche field, I've tried to gear this presentation to give, an overview first of what we do, but also then to focus on some topics that might be relatable for some of you who work in fields that are similar, at least in terms of the data being analyzed. So first of all, then I will give an overall introduction to concepts that I will use throughout my presentation, like what is a connectome. I will talk about connectomes, what they are. Then I will introduce our project, so the Human Connectome Project for disordered emotional states. And then I will talk about two studies more in detail, which are more methodological and I thought would be useful for a methods-oriented audience like yourselves. And then finally, I will close with some future directions of our work. So first of all, what is a connectome, right? I put it in the title of the presentation, but it's kind of a niche concept, so I'll introduce it here. So what we use in our research is a technology called magnetic resonance imaging. And, the way it looks, it's like a big scanner, like the one you see on the left. And thanks to this scanner, we can capture a lot of information about both brain structure and brain function. Brain structure, you can see it on the right, for example, how different parts of the brain are wired together or connected by bundles of axonal fibers. Or we can also measure how much gray matter or neuronal bodies are in each of the different regions of the brain, looking at the right. But finally, we can also measure functional measures using functional magnetic resonance imaging, or for short, fMRI. And fMRI is really an indirect measure of neuronal functioning or of brain function. What we really do is we measure the changes in blood flow on the whole brain in time. So for example, here at the bottom, you see a classical fMRI experiment where a person is in the scanner, they are looking at the flashing checkerboard, they can close their eyes or open them. And you can see here this bold signal or blood oxygen level dependent signal, which is what we're actually measuring in fMRI, is lower when people are keeping their eyes closed in the visual cortex. And then ramps up as they open their eyes, as the neurons in the visual cortex start to fire in response to this visual stimulus. And then it goes down again when people close their eyes, up again when they open them. So this is an indirect measure of neuronal functioning based on the blood flow. And this type of measures are what I'm going to talk about mostly today. Sorry. So again, what is a connectome, right? In general, we conceptualize a connectome as the way brain regions are connected together at the macro scale. And the interpretation of the word connected varies. If we're talking about structural connectomes, like the one on the left, it's literally the white matter axonal bundles of neurons that physically wire together different parts of the brain that can also be pretty far away. However, these are not really the connectomes I'm going to talk about today. I'm going to talk about the connectomes on the right, which are called functional connectomes. So the concept of functional connectomes is related to the concept of functional connectivity. So we saw that we can quantify the blood flow in different regions of the brain in time. And so usually what one means when one says two regions are functionally connected, it means that their time series mirror each other in time. So what I mean by that is you can see that, for example, the red region in this diagram and the yellow region, you can see their time series in the bottom part of the figure. You can see they track each other very closely. They're very similar. And so the conclusion one draws from that is that maybe their function is somewhat related. And we say in jargon that these two regions are functionally connected. Whereas by contrast, you can see the blue region, which is, completely different from the other two time series, even anti-correlated. We say that this region is not functionally connected to the other two. So the functional connectivity I'm going to talk about is this concept of like correlation of time series over large spatial scales. And, I mentioned we record the signal of the brain in time, right? And how this really looks like is, we scan the whole brain, and we can scan the whole brain pretty rapidly, about, even in under a second. And so we get a lot of data points, about 100,000 points in 3D space every less than a second. And some of our scans can go up to 20 minutes in length. So you can see immediately that this is a really large quantity of data to analyze. You can get up to 170 million data points per individual just by doing this type of scan. And so this is puts us in a situation which is very common now in very many fields of medical research, for example, genetics, but also if you're doing like neuropixel recordings from animals and so on. So we have a very high number of features, but our number of participants usually is relatively low because it's quite expensive to scan people in an MRI machine. And so we have to be mindful of all the problems that come with working in this regimen, such as overfitting. So how we reduce this data usually to make it more tractable is we slice up the brain into what we call brain regions or parcels. So which means like groups of points on the brain that we know are similar to each other because of their anatomy or their function. And so we usually then extract time series from those, like for example, by averaging them. And then we can have a reduced set of features. We correlate all of those time series together and we get a square, right? We get a square where each element represents the correlation between two time series of two brain regions. And obviously this square is symmetrical. And we call this a connectivity matrix. And usually that's how many studies deal with the complexity of this data. By doing this, people have observed that some groups of regions consistently correlate with each other more than they do with the rest of the brain. So there is some modularity in the brain networks you obtain in this way. And so people have started, identifying networks that consistently come up across studies. And you have some examples at the bottom, like a motor network, a visual network, the default mode network you might have heard about if you're in the neuroscience adjacent field, or the frontoparietal network. And these are all like pretty established networks you can identify in most people. So I'm part of the Human Connectome Project, which is a large-scale effort to map human connectomes, structural and functional. And you can see here, it's a pretty widespread project, many sites in the United States, also some in Europe. We're here at the very, very left of the United States in the San Francisco Bay Area. And our branch of the Human Connectome Project deals specifically with trying to study these connectomes in people with mood and anxiety disorders. So now that you all know what a connectome is and how we derive one, I'm going to talk a little bit more about our own branch of the Human Connectome Project. So I study mood disorders, and mood disorders are a significant public health crisis. They are the leading cause of disability worldwide. And one thing that people have observed when studying mood disorders is that normally clinicians use this manual, the DSM, which is a manual for psychiatric diagnosis. And the DSM really works by putting people into bins based on whether they meet some criteria or not. And then they get a binary diagnosis. Either they have major depression or they don't. Either they have generalized anxiety disorder or they don't, and so on. The research recently has been going into a slightly different direction. We're trying to capture, these symptoms of mood disorders more as dimensions, as continuous dimensions, right? Because maybe it's not exactly like a step function where someone either has anxiety or they don't, but there's a continuum of severity of anxiety that a person could have. And so the NIH has come up with a framework called the Research Domain Criteria Framework a few years ago, which is a research framework that has the goal of linking neural circuits, behavior, and self-reports, so different units of analyses, in a dimensional way to study mood disorders. So what we really do in our project is we use this framework, and we use imaging protocols derived from the Human Connectome Project, as well as behavioral tests and self-reports, to try and link them all together in anxiety and depression. So our project recruited 250 untreated young adults with symptoms of anxiety and depression, as well as 50 healthy controls. And we ran scans that were very similar to the other Human Connectome sites in the hope that one day we will be able to merge with them. And our goal really is to study, again, how connectomes, symptoms, and behavior relate to each other. And we focus in particular on some specific networks and that we hypothesize are linked to the cardinal symptoms of mood disorders. And these would be in the RDoC lingo, negative valence, which relates to symptoms of low mood, or anxiety, positive valence, which links to reduced pleasure or loss of pleasure, called anhedonia. And then cognitive systems that relate to the common symptoms of disturbance in concentration and cognitive dysfunction in these disorders. We also look at some cross-cutting dysfunctions that we link to some of the more famous resting state networks in the brain. And, our data will be shared with the research community. And, we try to focus on portions of the connectome that are more reliable. And we try to leverage other data sets from the big consortium to show that our results generalize. And I'm going to talk about these things a bit more in the upcoming slides. This is the RDoC framework that we're using. As you can see here, each type of symptoms, or, we have each type of domain, which are these RDoC constructs like negative valence. Within the negative valence, you have threat, which is more related to anxiety, loss, which is more related to low mood. And for each of these, we have a battery of imaging sequences, a battery of behavioral tests, and a battery of self-reports that we believe are linked. And we're trying to find link across these for each domain. And this is our study design, where we have healthy controls only doing a baseline visit, and clinical participants being followed in time every 3 months up to a year to just see how their symptoms evolve. These are the brain circuits we're focusing on. I won't get too much into detail, but I'm happy to answer questions on this. But basically, we hypothesize that each group of symptoms or behaviors will be linked to a specific brain circuit based on previous findings from the literature in mood disorders and meta-analytic evidence that these are networks that are specifically involved in mood disorders. When people are in the scanner in our project, we make them do some tasks. These tasks are a bit like a stress test would be for example, cardiology, when the people have you run on a treadmill just to see how your heart responds to that. So in the same way, we deliver some tasks in the brain that we know activate some regions whose function we're interested in. So for example, this task where you have to match either abstract shapes or match faces expressing strong negative emotion, we know activates the amygdala and other brain regions that are relevant for emotion processing. So we use this task as a probe of that circuit or that system of regions. And similarly, we have another task for what I mentioned before, for the reward circuit, which is involved probably in the loss of pleasure experience that people with mood disorders have. And in this task, basically, people can either win or lose money. And by seeing how their brain responds when they win money versus when they lose money, we can probe the activity of those brain regions that are related to the feeling of reward. And finally, we have a working memory task or continuous performance task where people have to constantly update their working memory and remember a letter they just saw and press when they see that letter again. And again, this task is designed to engage the brain regions we know are involved in cognitive dysfunction, in depression and anxiety. And finally, we have a lot of resting state data. The resting state is basically a recording of the brain in the absence of task data. So basically, it's supposed to capture how your brain just functions in a state of relaxation. We tell our participants to just let their mind wander as it naturally does, and just watch this cross on the screen so they don't fall asleep and keep their eyes open. But other than that, it's just a baseline, so to speak, acquisition. And a lot of our research also focuses on this, and a lot of biomarker research focuses on this type of scan. So I'm going to talk about this type of scan a bit more, because obviously it's easier to acquire than a task. And so a lot of people, when they do biomarker development, will focus on resting state especially. So our study was pretty severely impacted by the COVID pandemic, as many others. So we were late in finishing, but now we have finished. And we had all the data. The follow-ups are still being collected. Some people still have to finish their 12-month follow-up. And we're still really analyzing a lot of the data from this study. So today, what I'm going to do for you is, instead of focusing on our own data, we have done some methodological development, or at least we've tried to answer some of the methodological questions that we want to tackle, that are useful to tackle our real questions in our data set. And to do that, we have looked at some similar data sets that existed, and had been collected prior to the COVID pandemic. And we've used those to do method development or to answer some methodological questions, hoping to learn something that then we will apply to our data. So today I'm going to focus especially on this data set, which is the healthy young adult data set collected within the Human Connectome Project. This is a very famous data set that has been used for methods development in neuroimaging for a long, long time. It's a huge data set. It's about 1,000 controls that have been scanned a lot. So many tasks, very long resting state acquisitions, and also some repeated scans, which will be of interest for the subsequent sections. So our hope was to use this very large data set on controls, with only controls, to answer some methodological questions we had. And then what we learned from that, we can apply in our smaller sample of just 300 people. So the first study I'm going to talk about is, a study we did on test-retest reliability of functional connectomes, right? And so the rationale for this is there's been a lot of talk in the neuroimaging community about how reliable really are the measures that we can get from brain scans, especially functional brain scans. Structural brain scans are very reliable. They don't change much in time, but functional scans can be, sometimes quite unreliable, depending on what you're measuring. So ideally, we want a clinical biomarker to be reliable, right? And, what that means is if I scan you twice and your symptoms didn't change, or if I scan you twice very close in time, hopefully your brain measure should be similar at both times. I will spend a few words more on how we defined reliability here, but that's, intuitively a good thing for a biomarker. Also, there is another more subtle aspect of reliability, which is, when you try to do a correlational study or relate a cross-sectional measure to another one, for example, brain function and symptoms, the reliability of these measures really mathematically puts an upper boundary on the power you have to detect this relationship. And there's been many studies recently that have, bemoaned the smallness of effect sizes in your imaging. And I believe part of it is also because of this. In the literature, it wasn't very clear which connections in the brain, like which functional connections in the brain are the most reliable. And, building a connectome involves a lot of different steps. Definitely, there's a lot of degrees of freedom in terms of how you analyze your data. And so it wasn't very clear how these decisions would impact reliability of what you get at the end. And also, finally, the previous studies on reliability had relatively small sample sizes. And also they had these really long intervals between the scanning sessions where you can't really know if it's just a measure that's unreliable or if something really changes in the brain, right? So we wanted to use this large Human Connectome Project in healthy young adults to answer this set of questions regarding reliability. So first, identify the most reliable functional connections in the brain. Are some more reliable than others? And then assess the impact of some analysis decisions that that might decrease or increase reliability at the end of the analysis. And these are a bit like some niche nerdy neuroimaging questions, but I will go into them a bit more in detail. So the first one is the choice of the regions you use to build your connectome, right? The second one is a practice that some people do, some people don't, called global signal regression. And the third one is thresholding. So when I talk about, brain regions and how you choose them, I said before at the beginning that when we build a functional connectome, we slice up the brain in different ways to then build what is called a connectivity matrix, which is that square I talked about. And obviously you can slice the brain many different ways. Like some we call these atlases or parcellations. And some of these parcellations are based on brain structure, some are based on histology, some are based on brain function. So each of them is a bit different. And so obviously, depending on how you slice the brain, then your connectivity matrix seen on the right is completely different, right? Because the rows and columns will be different. So the output will be different. So the first question we ask is, is there a difference in how we slice up the brain? And is one way generating more reliable results than another one? Global signal regression, this is a controversial step that some people take and some people don't. So the reason for this is that most of the correlations in the brain are actually driven by physiological signals like breathing, heartbeat, and so on. So it stands to reason that because we're measuring a hemodynamic signal, when, for example, someone is breathing or heartbeat, they will have an impact on this signal and they will artificially inflate the correlations between the brain areas because obviously the heart is beating at the same time for all brain areas. So this will generate an artificial correlation in these hemodynamic signals. So some people, there's many ways to account for that. One thing many people do is they just remove with regression an average of the signal over the whole brain, which is supposed to remove all of these kind of autocorrelations that are generated by noise. And so you can see here the impact of that. On the left, you have a connectivity matrix without global signal regression. And on the right, you have it with global signal regression. And you can see that most of the artificially inflated correlations just go away. And what remains is this beautiful like modular architecture of the brain divided into networks, which is what the big squares on the diagonal represent. And finally, again, people in other fields do this too. When you do a connectivity matrix, you get a fully connected kind of graph. You can model it as a graph, like where the nodes are the brain regions and the edges are the strength of the connectivity between regions, but the graph is fully connected. So it's hard to make many conclusions on fully connected graphs. So people, what they sometimes do, they will threshold this graph and say, okay, let's just pick the top 25% connections, right? And then you get like a sparse graph, like the one on the right. And then you can actually do some, graph analyses on it. You can do graph metrics, compute some measures of, how strongly the nodes are connected. Is there a difference between clinical and healthy and so on? And you get a graph. But it is unclear, what happens when you do this regarding reliability. Is this a way to boost reliability? Like, are the regions that survive, are the connections that survive this thresholding more reliable or not? So that's what we wanted to see. So we used the data from around 860 healthy people, and each of them was scanned on consecutive days for an hour each day. So that's a lot of resting state data. So, this is kind of an ideal scenario. So where you have a lot of data on consecutive days where you don't expect the brain to change that much. And so, obviously, each connectivity matrix, depending on the atlas you use, can be pretty large. So you can go from around 20,000 connections to 60,000 connections. And we tried, to compare in terms of reliability, 3 different atlases, doing the global signal regression, doing different ways of thresholding. And then we compared also like individual edges of this network and also the mean of connections of those famous networks that people keep seeing, hoping that this would be a potential biomarker for mood disorder. Anyway, we compared around 10,000 connectivity matrices in this project. Now, a few words on how we operationalize reliability. We use something called the interclass correlation coefficient. And this captures basically 2 things. First of all, so there's 2 ways, two desirable properties of a biomarker you really want. The first one is measures from an individual should be, repeated measures from an individual should be as similar to each other as possible. And the second thing is measures across individuals or between individuals should be as different as possible. And this is kind of the ideal scenario. You can see it at the top right, where each measurement from the same individual is really similar. And each individual is very different from other individuals. And that's kind of an ideal scenario if you're doing correlational studies, because you can, both tell apart people and also you have a reliable measure within the same person. Of course, as these things get worse, it becomes harder. So if the measures you take from the same person are more and more different from each other, then it becomes harder to capture, these between individual differences too. And the same thing, if people become more and more similar to each other, then it's harder. And this ICC will basically capture both of these things into one metric. And you can see as the ICC drops, it's harder and harder to differentiate people. And there are some cutoffs for the ICC that are arbitrary, but they were convenient to bin brain connections into different bins and quantify how many are good, how many are bad, and so on. So looking at the results. So when we did this, first striking result is actually a very small percentage of connections is very reliable, has excellent reliability. It's about like 10%. 0.10%. That's still a lot of connections because we have so many, right? And the interesting thing is on the right side, you can see a plot of these connections on a brain. They're kind of always involving similar regions, like the dorsolateral prefrontal cortex, the superior parietal lobule, like this back part of the brain, and the temporal lobe. And so it seems that the connections between these regions are the most reliable in the brain, no matter which atlas you use. Good connections were around, 6 to 8%, which is still thousands. So it's not that bad, but there's still a small fraction compared to the total. And the median ICC was actually, not that high, around 0.41 to 47. So more than half of the connections in the brain are probably not that reliable. This is just a plot showing which nodes are involved in the good connections. And you can see, again, kind of this pattern showing up where it's like more often the dorsolateral prefrontal cortex, some parts of the parietal lobe, and some portions of the temporal lobe. Whereas other regions, they don't seem that reliable at all, like some of the motor cortex, sensory cortex, and also the subcortical regions, especially shown on the right, like, for example, the amygdala or the caudate nucleus and so on. Good news for the people working on biomarker development. If you take the famous networks that people keep identifying and you just average the connection within them, you actually get a pretty reliable biomarker. It always has good reliability. I think it's because averaging reduces some noise, and actually this helps in this case. So this is just a plot showing that if you average a bunch of famous networks that people know and use a lot in their research, you always get usually a measure that beats that has good reliability, which is 0.6 ICC. One interesting thing is that we observed that doing the global signal regression consistently decreases the reliability. And really, the relationship between the strength of the connection and the reliability is very complex. It's not linear, as you can see on the right. And this makes it so that if you threshold these matrices based on their strength, then you almost always lose reliability, because you're basically cutting off some connections in one session, but maybe you're cutting off another connection in the other session, and then it becomes very messy. And so in general, thresholding seems to have a negative impact on the reliability of the connections you get. So overall, from this study, we concluded that actually a small portion of the functional connectome has a good to excellent reliability. And the interesting thing is this frontoparietal network seems to be especially reliable. We've seen that averaging connectivity within known networks actually gives reliable biomarkers, which is good. In general, global signal regression decreases reliability, and thresholding is complicated. And I'm happy to talk about it more in the questions. In general, we hope that considering this information could increase power for studies that want to look at how brain networks, like functional brain networks, are associated with measures that vary across subjects. So with this, I'm finished with the first study, and I'm going to talk briefly about a second study we did, which again could be of interest for even fields that are outside of neuroimaging. And the problem here we were trying to face is, how do we correlate our functional measures, which are so large, with our symptoms and behavior? And many people, like many studies for this application, they used a technique called canonical correlation analysis, which is a very intuitive and simple technique, having two sets of variables. It will look for linear combinations of variables that maximally correlate. So basically, if on one side you have your brain measures, on the other side you have your symptoms, you want to find a set of weights for each of your features on each side of the equation, so that when you sum them together, you do a weighted sum on both sides for each person, you get two variables that maximally correlate. And many of you might be familiar with this technique. And so we really wanted to see if whole brain connectivity can be related with this technique to self-reports of negative emotion in daily life. And also we were interested in another slightly, more specific question, which is, we think that our tasks, for example, the emotion task I showed at the beginning, where you have to match those negative faces, we think that by probing the regions we care about, we can get information that's more relevant to map to certain symptoms. So we asked the question, is it easier to correlate rest or task connectivity to self-reports of negative emotion? When I tried to do this, actually, what happened is, I tried to do it, but I couldn't, because many methods that were out there to do this did not work on my data. The first thing is that the data we are using here has a huge number of features, and many algorithms will just crash. And the reason is that they're trying to build a giant covariance matrix, and then they would just crash if you have more than 1,000, 10,000 features. Another thing is that, many of the existing algorithms, they are very agnostic, and they don't incorporate any knowledge in how these correlations should look. Like we have already said that that brain networks will be more correlated within them than across them. And so we wanted to find a way to incorporate this knowledge into the algorithm and trying to take that into account when we were looking for, these correlations. And finally, many studies in the past have, used CCA, but CCA is extremely prone to overfitting. And so was really key for us to use an unseen test data set that, to avoid this problem. And this was a collaboration with some colleagues in the Department of Statistics, in particular, Dr. Tuzzi, who is a statistician who developed a new method to help us overcome these limitations, and Professor Trevor Hasty. So, we took a pretty large sample of people from the healthy young adult data set. And for each person, we had a large amount of resting state data, but also that emotion task that is supposed to probe areas involved in negative emotion. And so we did this analysis for both. And then we had a questionnaire asking them how much in their daily lives they feel these negative emotions. And you can see we have quite a few of them. There's anger, there's fear, there's sadness, and so on. And we tried to relate the imaging to these questionnaires. And this is, again, a depiction of the task that we use and the resting state. So the task is just a task where people have to match either abstract shapes or faces expressing negative emotion. And what people have seen is that this task elicits activation consistently in brain regions involved in negative emotion. So, for example, the amygdala, the medial prefrontal cortex, and so on and so forth. So, and on the right, you have your normal resting state. And so our hope was to see that, maybe the task is better than the resting state if you want to match what people feel in their everyday lives. So I won't go into the details of how we obtained the connectome in this project, but there's many steps in pre-processing to get to the measures that we care about. But what's most important, we got one of those big matrices for each person for the rest condition, and one of those big matrices for the task condition. And so here is where the algorithm that our colleagues in statistics have developed comes in. So there's two problems, as I said. So obviously we need some kind of regularization because the data is huge and it will overfit like crazy if we don't regularize. So we need the regularization. The problem is the existing regularized CCA algorithms couldn't handle so many features. And so our colleagues in statistics came up with some elegant mathematical tricks so that the algorithm can actually very quickly solve the canonical correlation problem, even when you have tens of thousands of features. It's actually surprisingly quick. It takes only a few seconds, and that's really great. And also, we had that other problem where we know that brain regions belonging to the same network will show correlated activity to each other. So ideally, we wanted our weighting scheme, like the weights that come out of the algorithm, to also respect those kind of functional boundaries between the groups of regions. And so the way we did this is we implemented a regularization procedure that that has two parameters. One parameter is trying to make the coefficients assigned to each brain network be as similar as possible. So each connection that belongs to the same network will be as similar as possible as this parameter increases. And another parameter is a regularization parameter that will try to shrink the whole network, the weights assigned to the whole network, to 0. So we tune these two by cross-validation, and then we test in a held-out portion of the data set, as you can see here. So basically, we split the data set in two, an unseen test set and a trained set. We do cross-validation on the trained set to find, the lambda and mu, which are those two parameters that maximize the correlation we find. And then we apply them on the test set. And because here you will see the results are really small, like really weak correlations, to evaluate if these correlation is real or not, we also did a permutation test on the test set to see if it beats like a null that we generated by doing permutations. So this is just the cross-validation. You can see that on the left, so each line corresponds to one value of one of the parameters, which is mu, and the x-axis is the other parameter, and the correlation on the left. sorry, on the y-axis. And you can see that at low values of the parameters, basically the CCA will overfit like crazy. You see that the correlation that comes out is 1, almost, almost always with small parameters. And then it starts to actually not overfit when you get to a pretty high value of the parameters. And the correlation on the test set of the cross-validation obviously goes up in a corresponding way. And this is the final correlation we found that generalized to the held out data. It's a correlation between our questionnaires and brain. And you can see it's pretty weak. It's around 0.175 weak, but still, beats the null. As you can see on the right, we did a permutation test where we just shuffled the rows of the original data, where we shuffled the original data a lot of times, I think 10,000. Calculated the correlations we would expect being due to chance. And we can see that our correlation actually is pretty unlikely in conditional on the null. So that gave us some confidence that this could be a real correlation. And, the challenge here was that, the pattern of weights assigned to each brain connection was pretty complex and hard to interpret. The only good thing is here, our algorithm, because of the way we implemented the regularization scheme, penalized connections that went across, brain networks. And so you see that you can kind of see that some of the blocks along the diagonal seem to be more weighted or have higher weights than the regions that are off from the diagonal. So that kind of helped us elaborate an interpretation for these data. And this is the same matrix you saw before, but this time averaged by network. And you can see that patterns emerge where, for example, the visual network seems to be strongly weighted, and other networks such as the orbitoaffective network, OAN, you can see there's a big red square there, and so on. And regarding the weight questionnaires, we found that this canonical component mostly involved feelings of anger and aggression in particular. So, at the end of the study, what we did was develop this new algorithm, which extends canonical correlations to model the properties of functional connectivity within brain networks that are established. And we used it to identify these connections that correlated with subjective negative emotion, but still the correlations for this specific application we detected were weak. One thing I didn't mention, apologies, is that actually the correlation we found was true only in resting state data. In the task data, we didn't find anything. And so we think that possibly in this case, resting state was more promising. We think that it's because resting state had so much more data. It was 20 minutes versus only a few minutes in the task. And so, and also like the component we identified seemed to have something to do with more of a trait, aggression, like more of a personality trait rather than individual episodes of aggression. So maybe, resting state, which is more like the baseline, brain baseline, helps more to disentangle that. Definitely one take-home message is that, correlation neuroimaging studies like this one, or any study really working on such a big number of features, really need cross-validation, regularization, and testing on held-out data, because the inflation of effects will be just huge. And this is no news, but it's good to reiterate that. More interesting maybe for many of the audience, GRCCA is actually available as an R package. So in reality, this can be used for any data that has a known correlation structure. So it just takes in like a set of labels kind of for each of the features, telling it with what other features it's expected to correlate. And then it takes that into account when doing the canonical correlation analysis. So really it can be used for anything, not just neuroimaging. So with this, I'm concluding my presentation, and I just wanted to give a big overview. So what we do at our center, we try to, use these large-scale brain imaging data sets to develop new biomarkers that relate to the symptoms of mood disorders. We showed that functional connectivity of brain networks is a candidate and biomarker for this purpose because it can be reliable. We need to really think a lot, I think this is my personal opinion, to think about reliability, and define it also in a way that matches actually what we want to develop. Like sometimes people care about how people will respond to a drug. Sometimes people care about, is the diagnosis reliable? Like, does my biomarker match a diagnosis made by a clinician? Can we predict symptom trajectories in time? And depending on that, definitions of reliability might change, but it's, I think, definitely something we should always keep in mind. Brain-wide associations are likely to be small. When you take the whole functional connectome and try to relate it to something else, you probably find small effects. There is this famous paper that came out last year by Marek et al. Saying that whole brain associations with other variables require thousands of individuals. That's kind of the headline of the paper. I think that reliability is also important. So that, like I said, it puts an upper boundary to what you can detect. So I would add to that, sure, you need thousands of individuals if your measures are unreliable, but maybe if your measures are a bit better, you might need less people. And finally, focusing on some circuits that we have reason to believe are involved in mood disorders might be a way forward to actually get to some useful biomarkers. And then in the future, we plan to pool our data, with all the other sites and to, do our analysis in the clinical sample, as we had said. We hope to combine also fMRI with other types of MRI, like structural connectivity, and then relate our measures to longitudinal trajectories or treatment effects as well. And we're kind of in the process of doing all that. So I'd like to thank the director of our center, Dr. Leanne Williams, who is also the PI of this project. Also all the people who have worked, here in the lab and have worked on the Human Connectome Project with me, and also our collaborators from the Department of Statistics and also from Washington University, who directly, made the studies I presented today possible. And I would really, really like to thank you all for your attention. Thank you very much.
Amazing. Thank you so much for that wonderful presentation. We are going to open the floor for questions now. If you have a question, please use the Zoom to raise your hand. All right, I see Abby, go for it.
Sorry, I stopped the share to see who's talking because I can't see. Yeah, thank you.
Hi, yeah, thank you so much for your talk. You talked a lot about how kind of analysis decisions can affect the reliability. And so I'm wondering if the order of tasks or kind of rest time between tasks might have an effect on like the strength of connections found or on the reliability.
That's a great question. So usually what people do when they do resting state is they put it before the tasks so that there are no kind of bleed out effect of the task on the resting state. Like the assumption here is, oh, maybe because you've just seen some graphic images or, maybe depending what the task is, right? Then you're still, maybe your brain is not really at baseline. I think like here in this case, the resting state was before the task. So I don't think there's that effect. Yeah, I don't know of studies that have actually examined the order, like how the reliability would be affected by the order, which I think is very interesting. In this specific case, I think most of the difference we saw was because just of the sheer amount of data we had. Like resting state super long, and you have like thousands of data points per individual, right? Whereas the task is much less. So I think it's only natural that with more data points, you have less noise. And so it's easier to do whatever you're trying to do in this case. But I think there's an interesting point, and thank you for suggesting it. I didn't think of it. That's great.
Excellent. All right. Aris, please go ahead.
Thank you, Leonardo. Very nice talk, very interesting material. Thank you for sharing. I have a couple of things to ask. So first of all, when you first showed it in your first slides, right, when you showed how you build these matrices, and you pointed out that if two like regions of the function of the fMRI, like they go up and down together, they correlate, it's a positive indication. But I was surprised to hear that with anti-correlate, you would treat them as if they're not somehow related. In my mind, typically, strong anti-correlation is also an indication of networks working correctly.
Yeah.
So together, I'm sorry. that's just maybe you can comment on that. And then on the study design that you mentioned, right? I'm curious. You pointed out how you probe the individuals with different tasks. I was wondering if you think that the fact that in the anxiety study, right, that individuals are medicated, I would imagine, right, for the most
they're
part.
Not. Yeah, they're not.
Oh, they're not. Okay,
yeah.
so this doesn't affect actually the function. Okay, that's good.
Yeah.
And then the other question is, if you think, that the wiring that you guys built, could it be affected by the strength? I mean, usually when whatever instrument you're using, right, there's sensitivity. So I would imagine that maybe in some areas the blood flow might not be as strong, although the effect might be strong. And I imagine maybe some of those things are not captured. What are your thoughts on that?
Yeah, so that's all great questions. So the first question about anti-correlation, definitely I agree. So I didn't say it very clearly, but usually what you do is like actually you just assign a number, right, to each element of the matrix. And some numbers are also going to be negative, right? So anti-correlated. And many of the networks in the brain have been identified exactly because they are anti-correlated. So for example, when I talked about the default mode network, people have observed that, the default mode network will change its pattern of correlations depending whether people are doing a task or they are, at rest. So it will be highly correlated when you're doing a task, when you're at rest. And then its correlation will drop or even become negative when you're not. So actually, the negative correlation, especially if it's strong, the anti-correlation is also a valuable source of information. And I think it depends really a lot on how you model this type of interaction. There's some, building the connectivity matrix with the correlation coefficient, to be honest, is the most simple way of doing this, clearly, right? But there's many other methods that might capture that information well too, where you will also identify, and it could be of interest when two regions anti-correlate or are decoupled in some conditions compared to others. That's definitely a great point, though, that generally when people say functional connectivity, though, they do mean that it's a positive correlation. But I agree that you're right. If two things are perfectly anti-correlated, then they do have some kind of functional relationship to each other, probably. So that definitely depends on the method you use. Second answer is yeah, they are unmedicated in this project. Sorry if that wasn't clear. So that's
that makes sense. Yeah.
And the third question was about using seeing if different regions that are physically connected are also.
Actually, that sometimes the strength of the correlation, I guess, can be affected by the intensity of the signal, right? So the blood flow.
Oh, sorry. Yeah, the intensity of the signal. Sorry, the intensity of the signal. Yes, absolutely. So I think that's why for some regions it's really hard to estimate reliably functional connectivity, especially the deep regions inside the brain, how they connect to the rest of the brain. Because when we do fMRI, usually the regions closer to the surface of the brain, you have better signal, just because of that where the coil is, the receptor coil is on your head. So that definitely is a challenge. I think that when you do whole brain correlation like this, purely exploratory, where you draw these big squares, then it becomes tricky to look at subcortical regions because they will stand out as having lower connectivity to the rest of the brain. So you have to treat them kind of differently. And there's many ways of doing that. But I still think that maybe the best way is if you have some strong model or some priors that will tell you, oh, from this region, I'm interested in this region, goes to this region because of actual physical constraints or tracing or other studies. That's probably the best way to look at those regions, to be a bit more careful and less data driven, I think. Yeah.
Thank you very much.
Oh, also one thing we looked at also is because of the physics of MRI, some cortical regions have bad signal too, like the ones closer to the air sinuses, the sinuses in the head. There's some air and it distorts the signal, and we showed that the connections involving those are the least reliable of the cortical connections too, which goes exactly to what you're saying. Yeah. Sorry. Yeah.
Fantastic. All right, Dennis. You are muted.
You hear me, right?
Yes.
Yeah. Thank you. Very, very interesting talk. I was wondering, so in genomics and in general data science, so there is this analysis of of matrices, basically trying to identify using the theory of random matrices the basically eigenvalues which are beyond what you would expect in a random, let's say, shuffled matrix. So based on what you described, many of the connections probably are just spurious, right? So basically the idea is that you take a matrix and you identify distribution of eigenvalues, right? And identify eigenvalues which are basically stand out compared to the null model. So I just wonder if in this area this is.
Yeah, it's a great question. I think in neuroimaging, this is not used so much. And I think this actually shows that we should talk more with geneticists, because I think many of the challenges are similar. So there's always a lot to learn. So I know, some people do that, I think. There is a kind of a problem with our data is also that the whole brain will be correlated anyway, like basically. So probably you will always beat the null. Because just by the fact that you're recording from like the whole brain, that is, and it's a hemodynamic signal, as I said, you can try to remove these like correlations that are just driven by heartbeat or breathing, but at some level, the baseline, like if you just look at the average correlation of the whole brain, I think it will always be more than zero. So I think you could beat the null pretty easily in that sense. In the study I showed, I guess we are also applying one more constraint to this problem, which is like, not only is the connection like better than the null, or is it pure noise, but we also want to capture that between subjects variability, which for our application is of interest, like because we want to relate the functional connectivity to something that varies between individuals, like the questionnaires, right? Then there's that additional constraint. You can imagine you could have a brain connection that's like super reliable because everybody has the same value, like it's super reliable, right? But then it's useless to us, right? Because if everyone has the same value regardless of their symptoms, then it doesn't actually capture that variance we want. So in a way, we were like adding an additional constraint to the problem in a way. And I don't know if similar in genetics when you look for.
I mean, the overall correlation would not be a problem. You'll get it. I mean, you'll just get it as a first principle component. I mean, it's not a problem. You'll just identify it as a first principle component. So it will be automatically removed. So in this analysis, what you're interested is, are there like non-trivial, basically eigenvalues which stand for some kind of intricate correlation between data. It's just interesting.
Yeah, that's great point.
Dr. Tozzi, I have a quick question. So I've read a number of brain MRIs before, and one thing that I think about when looking at your research, which is fascinating and very interesting, is the pre-processing. Because I'd imagine between the functional signals and the anatomical signals, one, there's a lot to do there. And second, I know we're short on time, but how generalizable is it to other systems?
Yeah, it's a great question. And, pre-processing is like a huge field within the field of neuroimaging. There's people who have super strong opinions about it. And there's a huge garden of working paths, as they say. You can make a ton of decisions, and each decision will, in fact, impact your result quite dramatically. And, there's a big study in Nature that has been done a few years ago, and they gave to the same team a bunch of neuroimaging data to analyze, the same data, to a bunch of different teams. I was also one of the teams. So it's like, and then they saw that they diverged quite a bit. And that's because of these decisions that one has to make. I think now, fortunately, we're entering an era where this is becoming better, in the sense that, for example, in our lab, we use standards in how to organize the data, and there's a format now called the BIDS standard, which harmonized how people were actually storing their data. And then on top of that, now there are standardized apps that run as containers that do the same pre-processing flow in one shot with minimal user intervention, so that the whole data gets through a reproducible workflow. So that's what we're using now. So we migrated to this type of systems to ensure, that preprocessing is consistent and at least can be compared, with people using the same container. But it's still an open issue as to which preprocessing is better. People have very strong opinions. I think personally, for me, being more reproducible is better, especially if you do biomarker development. We're not really a lab developing methods for pre-processing. We do pre-processing to then start our work, on the actual work. So for me, like if I can run it as a container and then just get the data on the other side, and it works really well. So I'm really happy about that. Yeah.
Amazing. All right. Well, thank you all for your wonderful questions. And thank you again to Dr. Tozzi for being a wonderful first speaker of the year. With that, we will conclude. Thank you all for attending. And just a reminder for everyone that next Monday will be the hybrid seminar. So come to PH 2200 if you can. Alrighty, everyone. Thank you so much. Have a great Monday.
Thank you for having me. Bye.
Thank you.
Thank you.